Wonderful. All right. So I'm Sarah, and I work in the Office of Surveillance and Epidemiology. I am actually on the Sentinel core team. So some of you may or may not have heard of the Sentinel initiative at FDA. And so I'm part of the core team that helps run that program. So today I thought I would present sort of an overview of the system, sort of what I call a Sentinel 101 for those that may not be familiar with it. And then I'm going to give 2 examples of some work we've done, just sort of highlighting different types of work we can do, and then for the FDA, you know, how it actually helped our mission, our regulatory mission. I'll leave some time for Q&A at the end, but if you have questions in between, you know, feel free to interrupt me. And if I don't, you know, if you put one in the chat or I don't see it, somebody just jump in and stop me. All right, so starting at the beginning, what is Sentinel? Sentinel is FDA's medical product active safety surveillance system. So we assess use of medical products, safety, and more recently starting to look into effectiveness. It's typically drugs, but we can do biologics and devices as well. We also not only do these routine analyses, they help develop the data, the informatics, the infrastructure to support, you know, future analyses as, you know, as the field progresses. We try to keep up as much as we can. The key components of Sentinel are a distributed network of data partners. So I know many of us are used to working in a single site or a single sort of database, and what Sentinel does is leverage a number of data partners, and I'll get more into that in a little bit. We use electronic healthcare data, so that's mainly claims and electronic health record data. We do use a common data model to standardize the data, and we do have a pretty sophisticated QA process to ensure data readiness when we need it. So Sentinel was actually created in response to a congressional mandate. So FDA, the FDA Amendments Act in 2007, required the FDA to develop this active post-market risk identification analysis system. So it required sort of 2 things. It said, you know, you obtain access to a number of different data sources, and it laid out, you know, claims, EHR, and you know, registry type data. And then it said establish the system, and it laid out benchmarks for, you know, the number of basically people that we had to cover by certain times to make sure sort of we were growing the system. Also within the FDA 2007, they required FDA to, or they allowed FDA to require sponsors, so medical product applicants, to conduct a post-market required study, or PMR. So you may or may not have heard of those. But in order to require that a sponsor conduct a post-market study to evaluate, you know, to further evaluate safety after approval, we have to justify that we can't actually do it ourselves. So either using FAIRS, the adverse event reporting system, or now this the Sentinel system, this active post-market risk identification and analysis system. So if we can do it within Sentinel, we actually now by law are required to do a study rather than require a sponsor do it. So another piece of the act related to Sentinel. So I mentioned in 2007 was the mandate and the actual, you know, because it takes time to get these things up and running. Sentinel was launched in 2008 with the mini Sentinel actual pilot program launched in 2009. We did meet the benchmarks that I mentioned in the prior slide of 100 million lives by 2011. I admit I was not part of the Sentinel team then. I was still in graduate school then, but they did meet the mandate. And then in basically from 2011 to 2016, they were growing the system, doing some analyses, you know, establishing a common data model and partnerships with data partners, creating these reusable tools, basically SAS programs, to conduct these queries. And then in 2016 was sort of a formal launch of Sentinel within the FDA's regulatory process. So before then, it had been used for ad hoc studies, but not sort of routinely implemented. In 2016, it sort of became a routine part of review processes for the FDA. In 2019, so last year, we awarded the third 5-year Sentinel contract. And as part of that, we kind of restructured Sentinel. So now instead of a single operating center, we have 3 centers. So there's still an operating center that helps us manage the overall Sentinel system. There's also an innovation center that's doing a lot of work on different methods projects to kind of expand our analytic capabilities. And then we have a community building and outreach center to really help us, make Sentinel a national resource and reach out to other stakeholders, outside of the FDA. So a couple of slides on what Sentinel looks like. Hold on, bear with me. Technical difficulties, there we go. Okay, so Sentinel data from some data partners actually starts as early as 2000. And this graph shows the number of unique patient identifiers. So I would say number of unique patients, but it is possible if patients switch data partners that they're represented more than once, at different time points. And so this represents sort of data that we have in patients over time. So you can see early on, up to 2006, we have a little bit of data, and then really some of our larger data partners, some national insurers, start contributing data. And so, a lot of our routine analyses will go back to 2008 or 2010. And then you see on the right side, we don't think that all of our data partners have complete data through 2019, obviously not through 2020. So that's why you see that drop off, but that will continue to grow over time. So if you put it all together, we have over 200 million unique patient identifiers or patients with at least one day of drug and medical coverage. If you only look at medical coverage, we actually have over 300 million. And that data is going as far back as 2000. So in terms of how many are active patients right now, if you were to do a study right now, we have over 70 million members who are currently accruing new data. Total, if you're looking at person years, almost 800 million. Not surprisingly, because patients, might have a lot of, more than one dispensing. We have almost 15 billion pharmacy records. I won't go through all of this, but we also have a mother-infant linkage, which is a relatively new capability within Sentinel, where we link mothers who have delivered a live birth infant with the actual infant records so that we can do pregnancy safety studies. And so we have over 5 million of those linkages. What does the age distribution look like? You can see here the outer circle is sort of more narrow categories. The inner circle shows broader categories. So you see about 19% of our of the Sentinel distributed database is the pediatric population. Over 50% is an adult population, and then 28% would be 65 and over. I should note that we do actually have 100% Medicare in our data, so we do have pretty good capture of the older age group. So I mentioned the Sentinel data partners. Right now there are 16 core Sentinel data partners listed here. They are a combination of national insurers, integrated delivery systems, and Sentinel works by maintaining a distributed architecture. So here, the figure on the right shows that each data partner actually houses and maintains operational control over their own data. This helps us sort of leverage the statistical power by aggregating data from multiple data sources and not just one. But it also sort of addresses concerns over patient privacy. You can imagine that different health insurers aren't going to want to share data, their data with each other, or even with a body like the FDA without maintaining control over it. So we actually only see aggregate results, and that helps us, sort of have the power to assess the safety of. I apologize if you can hear that. That helps us leverage large sample sizes to assess safety, for rare outcomes or even rare exposures, but also sort of assuage any proprietary or privacy concerns from our data partners. So I think I've mentioned where our data comes from. A lot of it are national health insurers or larger integrated delivery systems. We have a number of state Kaiser sites. So all of these are basically interactions that patients have with the healthcare system. And the data are maintained either by the insurers themselves or the healthcare providers. So it's largely claims-based, and there are some electronic health record data. So you can imagine if you have data from a number of different data partners, it's going to look different, be structured differently, have different data elements available within each site. So, how do you leverage and sort of combine all of that data? The main way we do that is by leveraging a Sentinel common data model. So this is what our common data model looks like. I know there are a number of other common data models, and the FDA also has done projects to sort of map the different common data models. Each has their own strengths and weaknesses. So within Sentinel, I would say a large majority of our studies use the top row. Administrative data is typically what you would see in claims data. It's information about the patient in terms of enrollment and demographic, and then you have their drug data from, dispensings from outpatient pharmacies, encounter diagnoses and procedures from their medical encounters. And we also newly have a prescribing table for prescribing records from electronic health records. There's some other tables toward the bottom that are leveraged on specific queries that are not necessarily used in all of them. You can see, I'm going to highlight the mother-infant linkage data, which I mentioned earlier, which does link mothers to their infant records so that we can do pregnancy safety studies. So that's a really unique feature that we're using increasingly. Many of you may have either are currently in data or have been in data before, and so this might look familiar. It's like the kind of data that you might be working with. This is just an example of how data would be structured in the Sentinel Common Data Model. This is a subset of the tables that are available. I mentioned, demographic and enrollment. You can see the single person has about 10 different dispensings. From one encounter, there's a number of different diagnosis codes and different procedure codes. And so these are the data that we are using in our studies in Sentinel. And in terms of data quality, for the FDA, for any study, you want to have the highest data quality available, but really to be able to make regulatory decisions that impact, the public, we have to make sure that we have the highest quality data. And because a lot of these questions arise and where we need answers fairly quickly, we want to make sure that data are ready for use, as soon as the question arises. So as data are refreshed, so each data partner will refresh their data, adding new data either quarterly or biannually or annually, depending on the partner. Every time they refresh their data, they undergo this sort of quality assurance process. And there's multiple steps involved. I'm not going to go through it in detail, but there's a lot of different steps. They go back and forth at the operations center when some data don't look correct. And here, every time it goes through this quality assurance process, it's actually the whole data. So say they're refreshing data and adding 6 months. It's not just the new 6 months that undergoes the data quality, but the whole, the whole bulk of their data. I know in the past, anecdotally, we've had a data partner transform their data, and a year of medical encounters was lost, from a prior year, even though the new year looked good. And so when they look at sort of the trends over time as some of the data quality metrics, they were able to see that and go back and correct it. And so then when data are sort of up and running and made available for use, we know that as soon as we are ready to run a new query, the data are sitting there. We don't have to spend time sort of making the data ready for use because that's done prior. I know personally, when I was doing my dissertation, I recall it took me 9 months to get the data cleaned and ready for use, and only actually about 3 months to run the actual analysis. So this makes sense to me in terms of making sure that the data are ready for use, because it is a pretty burdensome process. Switching gears a little bit. So I'm going to talk a little bit about ARIA, which is our Active Risk Identification and Analysis System, and it's really the core subset of Sentinel. So if you remember back to the FDA, the FDA Amendments Act that I was talking about, it required FDA to establish an active risk post-market identification and analysis system. And that's where this term ARIA comes from. And there's a lot going on within Sentinel, but we call ARIA the combination of the routine analytic tools, which are these predefined parameterized tools that allow us to conduct safety surveillance or programming more quickly, in combination with data from the common data model. So when I say Sentinel analytic tools, we group them into sort of larger level categories. So focusing on level 1 and level 2, these are the bulk of our analyses. Level 1 you can think of as any sort of descriptive, univariate or bivariate analyses, unadjusted. We have a query builder tool, which we use internally to run a really, really quick analysis of just drug utilization. Level 2 are what you think of as sort of your adjusted analyses, your multivariate, where you adjust for confounding. We have a couple of different capabilities within that. So going back to signal identification, this is where we don't necessarily have a specific safety signal or safety concern in mind, but we say, this is a new drug, a new product, the first one in the class. We don't really know what to expect with it. We're going to sort of look across the board very broadly and see if anything, has an unexpected rate of, a certain adverse event that we can then investigate further. So signal identification, that tool, we typically right now use tree scan, but we're developing other capabilities there as well. Those would be the signal identification tools. And then level 3 is a sequential analyses, which is basically level 2 repeated over time. And this is when you need, you're sort of monitoring prospectively, as data accrue. So how do we use ARIA? We use it actually both pre-market and post-market, largely post-market, but going back to the FDAA mandate, when new applications come in and the FDA is reviewing them for potential approval, I mentioned that we have to assess whether or not Sentinel, or specifically ARIA, is sufficient to actually conduct the study. Because in order to require that the sponsor conduct their study or a PMR study, we have to make sure we can't do that in ARIA. The study then itself would actually be conducted in the post-market setting, but we do have to make this decision pre-market or pre-approval. So moving to post-market, which is where the bulk of the Sentinel analyses are done, anytime a safety issue arises or another regulatory question comes up, we can do it. So newly identified safety signals, which previously were called tracked safety issues, TSIs, real world use questions. So if our review divisions want to understand, how are other drugs in the class being used, or, is this drug being used in pregnant women? We can answer very easily drug use questions, looking at potential medication errors, generic drug equivalents. We've been working recently with the Office of Generic Drugs to try to address some of their questions. Looking at REMS, risk evaluation mitigation strategies, and sort of before and after implementation of REMS, or how often are people getting certain monitoring. There's a number of different questions that we can use ARIA to do, and not just sort of the typical safety question, to provide context around literature or a sponsor submission. So ARIA is being used in a number of ways. So I do want to plug that we try to be as transparent as possible, and so we have a Sentinel website. We post pretty much everything. So the common data model and all of the documentation and the analytic tools, and basically the SAS programs, all of that documentation, they are publicly available on the Sentinel website. The ongoing Sentinel analyses. So once we've initiated an analysis, we actually have to notify the sponsor as well as part of our mandate. And so we post both the analytic package and the results so that anybody, including the sponsor, can take down that analytic package. If they disagree with some of our design decisions, they can modify some of the parameters and then rerun it in their own data. And then all of our analyses, even the descriptive, very simplistic drug utilization queries that we run, all of those results are posted on the website, basically for anybody to see. So let me get a time check. Okay, so how have we actually used it by the FDA? What actions might we take? So probably the most obvious ones might be, a label change, adding new information to a label, or a drug safety communication that goes out to healthcare providers or patients. Sentinel analyses have also been used to support advisory committee. There have been a number of presentations at advisory committees, including that included Sentinel data. Citizen petitions that are submitted to the agency requesting information or requesting the FDA take an action. information from RA analyses has been used to respond to those. I mentioned, use in pregnancy. So this has happened several times where, a sponsor is required under a PMR to do a pregnancy registry, and they have trouble enrolling patients, and they ask the FDA, we can't enroll patients. Can we cancel our study? So sometimes we'll do an analysis and say, what does the use look like in pregnant women? Are we actually seeing use? And so we've helped inform those decisions on whether or not those registries are actually feasible. So ARIA sufficiency, I mentioned, when we have to evaluate that, we conduct feasibility assessments, sample size calculations, things like that, to see if we would be able to do things in Sentinel. We've had a number of inquiries from other U.S. agencies. Recently, we've been working with CDSE. We've had inquiries from GAO, ASPE. We have a number of partnerships with other agencies, and we help work there. And then the hardest one to capture, but probably the most common, is no action indicated. So it's not interesting, but sometimes there's a safety concern, and we will analyze it, do an analysis in Sentinel, look at literature, and then we say, what, this isn't really that big of a safety concern. We think, our current labeling or the current sort of, risk mitigation strategies are sufficient. We're not going to do anything. So that in itself is an important regulatory decision. It's hard to sort of capture that or publicize that, but it's a valid regulatory decision, based on scientific evidence that I would say happens fairly often. So just to highlight, how we are trying to be transparent, this is a snapshot of a couple of websites or web pages from the Sentinel website showing how we have used Sentinel to make regulatory decisions. So the left shows lists all of the completed ARIA assessments. These are mostly inferential assessments, but some are descriptive. And if you click the arrow on each of those on the left, it'll give you a sort of drop down with a description of what regulatory decisions or actions FDA took, as well as a link to the results that I mentioned earlier. A PDF of basically all of the output and the data. On the right is just a different page, sort of highlighting mostly advisory committee meeting presentations that have incorporated Sentinel data sort of as anecdotes of how we've used the information. So let me pause here before I go into a couple of examples. If there's any questions on Sentinel itself or our work or how it's developed, I'm happy to take those now.
Hi, can you hear me? Hi, it's Annalisa Rubino from AstraZeneca. Thank you very much. This is a very informative presentation. Can I just ask you to go back to the point you raised at the beginning regarding, when is the FDA rather than the sponsor running the analysis, specifically for pregnancy, for access to the pregnancy linkage data?
So for any observational PMR, and I clarify that it has to be observational, because if it's a clinical trial, then clearly observational data is not going to take the place of a clinical trial. But for any observational study that the FDA is considering asking for of a sponsor, FDA has to sort of do an internal assessment of ARIA sufficiency, of whether or not ARIA or Sentinel is sufficient to address the question. Most pregnancy studies to date have not been we've not been able to do within ARIA because a lot of them have been signal detection studies. And so applying our signal detection tools, which are fairly new, on top of the mother-infant linkage, right now actually it's still methodologic development, but we are working on it. And so sort of the combination of the tools and the data has not been there for us to do pregnancy studies, but there have been a handful of studies that are either in progress or completed where there was a specific safety outcome, say, congenital cardiac malformations, where we have used Sentinel or ARIA to do a study rather than requiring one of a sponsor. Does that address the question?
Yes, thank you very much. Sorry, I was on mute.
No problem. Any other questions?
Hi, this is Tung Chan. I'm from AstraZeneca as well. Just to build on Annalisa's questions. So if the sponsor industry would like to conduct a study, like a PAS study, post-licensure safety study for product for, say, pregnancy outcome, that be possible using Sentinel, or can you clarify that?
Yeah, I mean, it like any good epidemiologist, I'm going to say it depends. You know, there are different components that go into the assessment of RA sufficiency, and I don't think I have the reference in here, but there's a guidance on, you know, on PMRs basically, and RA assessments evaluations are part of that. There's different components to the assessment. You know, can we capture the study population? Can we capture the exposure? Can we capture the outcome? You know, a lot of times there isn't necessarily a validated algorithm, or we don't think we can capture it well in claims. You know, can we capture all the key covariates? Sometimes, you know, if you need lab data or BMI, that can be tricky in secondary data. You know, and do our tools handle it? So it's going to depend on every study that comes up of whether or not you can do it. So I can't say across the board, all of these yes, all of these no. But you know, depending on all of those components, it would certainly be feasible.
Yeah, I understand it has to be feasible, but my question is more about if we would like the Sentinel team to conduct a study, then there's a mechanism for the industry to participate. I mean, to outsource this to some group.
I see, industry participation. So that's a hard question, and that actually has changed over time. So right now, industry can access Sentinel through the IMED program, which is part of the Reagan-Udall Foundation for the FDA. It's a program within that, and it allows you to contract with the Sentinel Operations Center, which is, you know, the same Sentinel Operations Center that we use. And so you can leverage, you know, the Sentinel system tools and the data that way, but it would have to be sort of an independent study. You know, we actually wouldn't see it. We might know that so-and-so is conducting a study, and that's all we would know. So there is a mechanism for industry to access Sentinel, but it wouldn't necessarily be in collaboration with the FDA. Right. I think that line has to be there.
Thank you.
Mm-hmm. And if you need to, I can follow up with some links to more information about this. I don't think they're in the slides, but I'm happy to provide sort of the resources on more information where to get that.
Great. Thank you.
Mm-hmm. Any other questions? All right, I'm going to go through two examples. This is by no means sort of encompassing everything we do, but two examples I thought were interesting. So I admit I did not lead either of these analyses. So my knowledge around the edges, especially on some of the background, is a little hazy. So bear with me here. So phosphodiesterase type 5, PDE5 inhibitors, sildenafil, tadalafil, are approved to treat pulmonary arterial hypertension and erectile dysfunction specifically in med. So the thinking in terms of the mechanism of action is it may improve uroplacental blood flow and early onset fetal growth restriction in affected pregnancy. So there was a set of trials. It was called Strider, and it was actually an international consortium of four randomized controlled trial sites. And they looked at whether maternal treatment, so treatment of the mother with sildenafil, can improve pregnancy and birth outcomes. So in middle of 2018, the Strider trial, one of the sites, the Dutch site, was actually terminated early because they observed excessive neonatal mortality. There were, I think, just under 200 women enrolled at the time. There at the second site in Canada, it was stopped soon after, although the other two sites in the UK and Australia and New Zealand actually didn't observe this. So there was sort of further investigation needed. There's a planned meta-analysis. And the question sort of that the FDA faced was, well, what's going on with the PDE5 inhibitors? And so they decided to do, because there was sort of a randomized controlled trial, they didn't want to necessarily replicate the trial, but they wanted to see in the US, what does the utilization look like? So this particular query, they wanted to assess the prevalence of PDE5 inhibitor use among pregnant women, as well as reproductive age women in general, and as well as look at some of the indications for use. So here they looked at women 15 to 58 years of age, and they looked at live birth delivery. So I will say the mother-infant linkage that we have looks really only at live birth deliveries right now. There are methods projects to expand that. Here they looked at a broad time range from January 2001 forward to March 2018. There has been a lot of work in pregnancy done within Sentinel. So now there's a validated algorithm based on diagnosis codes for pre and post-term deliveries. And here they use data from 16 Sentinel data partners. So they looked at 4 different PDE5 inhibitors listed here, and outpatient dispensings were identified using NDC codes, which is typical of pharmacy dispensing. Here they looked at PDE5 inhibitor use during live birth pregnancies, both overall by the 4 individual products, over time, so by year of delivery, by mother's age, and by trimester. In terms of indications, they looked in the 90 days before pregnancy all the way through delivery, so during sort of a patient's entire observation time, using ICD-9, ICD-10 diagnosis codes. They looked both at on-label and potential off-label indications for which these drugs might be used. So this, sorry. There we go. So this slide shows on the left the 4 different PDE5 inhibitors as well as overall, and then the columns show the different timing of use. So you see use in the 90 days before, overall anytime during the pregnancy, and then by trimester. So the first thing you can see is that most of the use of the 4 is sildenafil in the US. And then the second thing you can see is that most use decreases substantially after the first trimester. So here you can see 2.6% per 100,000 live births in the first trimester, and then that drops to below 1 for the subsequent trimesters. Looking at potential indications, you can see the distribution here. People could definitely have more than one indication, so that's why these would not add up to 200%. So overall, so we see 2/3 of these prescriptions had actually more than one diagnosis of interest. We see a lot of reproductive diagnoses and related, including preeclampsia, fetal growth retardation. You do see a fairly high number for pulmonary arterial hypertension. So this is actually really useful information to the safety team that was evaluating the safety issue. Overall, FDA found that this is fairly reassuring. So they found a pretty low prevalence of PDE5 inhibitor use in pregnant women, at least in the US. And so, sort of taking that into account with the data that kept coming out of the Strider trials, as well as data from other sources, they, at that time, they decided that no action was needed. I'm not sure, I admit I don't know if they're still following this now, or what has happened in the last year. So if you're going to ask that I don't know. But I guess that was sort of an example of no action needed, in using Sentinel as one piece of evidence, in a body of work. I know Sentinel has rarely been used as sort of the sole piece of information, but it is often leveraged in combination with things like adverse event reports. Here there's data from trials, data from other countries, things like that. So I'm going to move on to this second example, which might be a little more in depth. So So this example is on hydrochlorothiazide. So it's a diuretic that's used to treat hypertension. It is given as monotherapy, but it is fairly commonly given in combination with other hypertensives, other antihypertensives. So several years ago, there were a couple of observational studies in Europe, I think mostly Danish studies, and they found an elevated risk of non-melanoma skin cancer associated with hydrochlorothiazide use. In terms of the mechanism of action, thiazides have been shown to have potential drug-induced photosensitivity, which can result in developing cutaneous diseases if you have exposure to both the drug and light. So the Sentinel team, or the Sentinel was asked to assess the risk of non-melanoma skin cancer for patients treated with hydrochlorothiazide and all related products. And they decided here to compare it to angiotensin converting enzyme inhibitors, or ACE inhibitors, and all products containing ACE inhibitors. So in addition to assessing the risk, they also looked to see if there was a dose-response relationship. And I'll show you that analysis shortly. So this was a retrospective cohort study. They used data from, all the way back from January 2000 to get as much data as possible, and because hydrochlorothiazide is a fairly old drug. Here they leveraged data from 17 data partners. So their exposure, they looked at new users, so incident users of hydrochlorothiazide from any hydrochlorothiazide containing product, and compared it to ACE inhibitors from any ACE inhibitor containing product. They identified, similar to the prior analysis, they identified use via outpatient dispensing using national drug codes. In terms of the outcome, non-melanoma skin cancer was divided into basal cell carcinoma and squamous cell carcinoma. And these were identified using an algorithm, a published algorithm based on ICD-9 and 10 diagnosis codes in combination with procedure codes and/or NDC codes for a topical chemotherapy. They looked at incidence rates of basal cell carcinoma and squamous cell carcinoma, as well as relative risks overall, and stratified by different demographic characteristics, as well as cumulative hydrochlorothiazide dose. They did propensity score matching to adjust for confounding. And then I mentioned the dose response analysis. So a patient's cumulative dose category could actually update over time, as they continue to take hydrochlorothiazide. So looking at the data, we found over 5 million hydrochlorothiazide users that were able to be matched in one-to-one matching to new users of ACE inhibitors. I'm not showing the results here, but they were fairly well balanced after propensity score matching. You see here, the results are shown separately for basal cell carcinoma and squamous cell carcinoma, presenting the incident rate, both for hydrochlorothiazide cohort and ACE inhibitor cohort, and then the hazard ratio. So you can see that, So overall, you see there was not a difference in risk between hydrochlorothiazide and ACE inhibitors for basal cell carcinoma. You see a very, very slight increased risk for squamous cell carcinoma. If you can see my mouse over here on the right. But then when broken down by race category, there were some interesting findings. I think I might have an older version of the slide. Oh, no, I do. Okay. So if you look at the different racial categories, you actually see among Caucasians that hydrochlorothiazide was associated with small but significant risk, increased risk of both basal cell carcinoma and squamous cell carcinoma. You see a couple other race categories also for basal cell carcinoma also had an increased risk. But here the team, given the sample size, I think decided to focus on Caucasians. And this slide shows the cumulative dose, or the dose response analysis. So on the left, you see squamous cell carcinoma. On the right, you see basal cell carcinoma. My apologies for switching them from the prior slide. So here you do see an increased risk of squamous cell carcinoma at higher cumulative doses, but you don't see the same increased risk for basal cell carcinoma at higher doses. So basically, they consolidated these a little bit further, and basically above 50,000 milligrams in terms of cumulative dose is when the increased risk with squamous cell carcinoma was seen. So in summary, the lack of an increased risk of basal cell carcinoma and the very small increased risk of squamous cell carcinoma in the overall population, the study team felt was overall likely due to the low proportion of patients exposed to high cumulative doses. They felt that the stratified analysis was more correct, but it was consistent with previously published studies. Looking specifically among Caucasians, hydrochlorothiazide was associated with small increased risk of both non-melanoma skin cancer types. And then the risk of squamous cell carcinoma did increase at higher cumulative doses. So there was a dose response relationship for that, but not for basal cell carcinoma.
Hey, Sarah, I have a question or two. This is Caleb.
Sure.
But I don't want to disrupt your flow either. Should I hold it or?
I'm almost done. I have like 2 more slides, so.
Okay. I was just wondering, in these studies, it's really interesting. And I wondered about, let's see, for my own organization, I put in a question. Oh yeah. So, well, I mean, 2 things. So one was about healthcare utilization. So just the potential risks of, I don't know how close you are to these studies, but I guess I was just wondering whether the teams grappled with the potential that, you know, that people that are being treated are more likely to have healthcare encounters and therefore more likely to get referred to a dermatologist and have the opportunity to be diagnosed with a cancer. And then I guess, like related to that, like I wondered if you guys thought about negative controls where you could look at some other condition and show that there's no difference where you wouldn't expect one between the thiazides and the ACE inhibitors.
So I like I said before, I did not actually conduct these particular studies as part of the Sentinel team, and I can get into this if folks are interested. I don't right now conduct a lot of these regulatory studies, but epidemiologists in our review divisions do. I sort of help manage the program. I oversee the studies, I track them. So I'm aware of them, but I'm not necessarily intimately involved with them. So I'm not sure that I can answer, I'm the best person to answer your question. In terms of healthcare utilization, I'm pretty sure that's something the review team, the team incorporated. I'm trying to recall if I remember seeing that in the results. I know we routinely look at the healthcare utilization sort of as a marker of, you know, health-seeking behavior. So I would think that they sort of tried to account for that. I guess, would you think that it would be different for hydrochlorothiazide users, for ACE inhibitor users? I'm not sure it would be largely different, but, you know, that's something that should be looked at. So I don't know.
Oh, I see. Yeah, no, that's a fair point. Yeah, no, that's a fair point, because the comparator you would think would have
is an active comparator,
yeah.
Right?
Yeah. Okay. Thank you.
No, but you're absolutely right, and I'm saying now that I hope the study team accounted for that. I know. So they actually just had this publication accepted, and I think I need to update the slide to add that reference because I think it was just accepted last week. But hopefully, you know, you can read it yourself as well. Yeah. I'm sorry. There's a second question.
Well, the temporality and negative controls. I mean, it would be nice to see analyses of temporality, because you wouldn't expect these cancers to pop up in 3 months, right? And then, so I don't know the length of time that they used. And then negative controls also, like all of these are just different, you know, variations on a theme that help to give one, you know, as much confidence as possible in the associations that are being described, right?
Yeah, absolutely. So starting with the negative controls, I mean, this particular study did not have one. I know that topic has come up fairly, recently a couple of times within Sentinel on various studies. So I know it's of increasing interest. I actually was just involved in, it was more of a method study with, it was actually with our Office of Counterterrorism and Emerging Threats, looking at flu sort of in preparation for a public health emergency. Imagine that. And we actually did a method study looking at, negative controls, but it was sort of in a flu context. So I know that method, that analytic sort of method is of increasing interest, but it was not used in this particular study. But that is a good point.
Thanks. And I think Mara also may have had one comment for you, if time permitting. Mara, go ahead.
Sure. Hi, thank you again, Sarah. That was a really great talk. I've been really interested to see it and see how far Sentinel has come. I was just wondering if you guys would look at effect measure modification to identify certain subgroups that may be at elevated risk from certain medications. I think your last example on hydrochlorothiazide would be a great example to look at that when you stratified by race, but I didn't see the interactions.
It's going to vary from study to study. And again, I wasn't part of this particular study. So I don't think that it doesn't look like they, now I'm blanking. I'd have to go back and dive more into the study in terms of effect modification.
Another level of evidence that you could do it with the causal inference methods there. So
yep. Yep.
Excellent work. This has been really exciting to see.
No, it's a good point. It's not something that we do very commonly, but obviously it's doable. I will say, in terms of translation, one of the things, like we have, the epidemiologists working on these studies, but then translating it, like you all have to translate it for, a larger, broader audience, for us to, translate it to medical officers or the folks doing, the product reviews, or issuing the post-market studies. It's always challenging to communicate these, these epidemiologic concepts to those, to folks that aren't trained in epidemiology. So in a different way, I think we have that communication challenge too. And I think, the sort of effect modification is one of those challenging concepts. So I know sometimes there's, I don't want to say pushback, but less interest in it. But no, it's a good point. And I can bring that back to the team that did this to see, sometimes even when they answer a question, they'll do subsequent work. So, if they are doing that, I can definitely bring that to them. Thank you. Let me just finish, and then I'm happy to take additional questions. So I think one of the, we on the Sentinel team were very happy. I think everybody in that group was very happy here, because this actually led to a label change, which doesn't seem like a big deal. I remember when I first joined the FDA, I said, okay, it's in the label. Who reads the label? Well, people do read the label. Maybe not most people, but people do. And we actually got a label change here with the results of the Sentinel study, which was kind of a big deal to have the actual Sentinel study sort of cited in a label. Let's see, yeah, I highlighted it down here. So this is sort of Sentinel use in action, basically summarizing the study. And but to my point earlier about communication, summarizing, the study was actually multiple analyses, a lot of back and forth. It was a couple of years, to summarize it here down to one or two sentences is, you can imagine, probably one of the hardest things for scientists like us to do. So just stepping back to summarize what Sentinel is, it is now, we consider it a mature safety surveillance system, semi-automated in that the tools are sort of available for use. So we use a common data model, not fully automated because it still requires people to design and conduct the analyses. It is now integrated into our routine regulatory processes, in a safety signal needs investigation. Even though it was mandated in 2007, and it's now fully functional, as of several years ago. The active risk identification analysis system, or use the ARIA, is the core really of Sentinel, using those analytic tools, a common data model that I mentioned previously. We really do try to keep our website. We've spent a lot of effort trying to keep the website useful and up to date. It is updated daily with sort of ongoing and completed assessments. So ideally, if you're ever looking for something about Sentinel, you can go there. We're trying to make it a good resource. And really, Sentinel is intended to provide information to FDA about real world data. How are our medical products being used? What is their safety and effectiveness in the real world? In order to help the FDAs with our decision making about our regulated medical products. So I will stop there. I'm happy to take any questions about Sentinel, these analyses, or anything else of interest.
Outstanding. Thank you, Sarah. That was terrific. I'm taking over as MC for Jody, who's off saving lives, or at least prolonging them with quality as her general internal medicine practice allows. But thank you so much. Yeah, we do have a few more minutes, maybe 5 minutes at least. I suppose maybe we can go slightly over the hour, but we won't keep you long. But please, people, feel free to add questions either in the chat box or come back online so we can see you and see you by video. And please, now's a good chance for you to get unvarnished thoughts from an expert within the FDA.
Hi, this is Chung Chen again. Can I ask a question about all these pregnancy-related outcomes? Are you using the ICD diagnosis code for those, or ICD-9, ICD-10? Or is there ways to validate some of the diagnoses, given they're very rare? I think they have a big impact on the analysis.
So in terms of pregnancy-related outcomes, I think right now, actually, we're finishing up some work to validate stillbirth as an outcome. For example, there's ongoing validations in both pregnancy and non-pregnancy. A lot of times we'll look into the published literature and use published algorithms that have been validated somewhere else. I guess, are there additional questions for?
So basically, you are developing algorithms to diagnose or identify those outcomes, right? It's not just ICD diagnosis, but some kind of algorithm. So.
I mean, again, it's going to depend on the outcome. Some will use just based on diagnosis codes. Some might use procedure codes. I know that there is a stillbirth validation that's ongoing right now, and it's using a combination of diagnosis codes. And in pregnancy, actually, the ICD-10 codes are much different than for ICD-9. They're much more granular. There's, weeks. Oh, now I'm blanking. I was just listening to a seminar on this last week. They actually had a two-step algorithm, first based on diagnosis codes, and then sort of adding either additional diagnosis type codes or maybe procedure codes. I can't remember. But I would say, if we typically, like most folks, look to literature to see if, what's been done in literature. If something has been done in an ICD-9 era, and this is not just for pregnancy, but for any outcome, we often do a trend analysis and sort of plot the trend of the algorithm in ICD-9 with ICD-10, and sort of, if it's a relatively flat, trend, then that gives us at least a indirect sort of, not validation, but confidence that the codes are sort of identifying, the same thing, without doing a formal validation. We can only do so many formal validations because they're not, they're not cheap and they're time consuming. But we have done some.
Yeah, thank you.
Great. Other questions folks have?
Hi, it's Annalisa Rubino again. If we have a couple of minutes, then I would like to ask you regarding the propensity score matching and the potential for residual confounding due to an imperfect matching, not because it was not conducted properly, but because we know that the choice of a specific treatment is linked to the profile of the patient, and often that profile cannot be fully captured in the databases. So it's difficult to put a threshold, but would you really think that, you know, an incident rate ratio 1.17 represent an excess risk, or there is a potential for residual confounding even when the methodology is properly applied and you know, all the statistics is correctly conducted?
So that's a hard question. I think that's where a lot of scientific and medical judgment comes in, and that's a conversation that the epidemiologists would have had with the medical officers that were, you know, familiar with the product and that therapeutic area. So I honestly don't think that I'm qualified to address sort of whether or not that risk is meaningful. In terms of propensity score matching, obviously we know there are limitations. There are other methods available in Sentinel. There's, you know, self-controlled designs available, you know, depending on the study question. And recently, actually, we don't have now just propensity score matching, but a lot of sort of a broader range of propensity score based methods are now available in Sentinel, you know, IPTW, you know, risk stratification. But you're right, you know, a lot of it goes down to what the data you have are. And we know that secondary data does have limits. And so that sufficiency assessment that I was just referring to in the beginning of, you know, can we study something in ARIA? It's sort of, can we study something using observational data? And sometimes the answer is no, you know, depending on, you know, if you know that, you know, BMI or smoking is a big confounder and you can't capture it in your particular data set, you know, you really shouldn't do the study. And we have made decisions, you know, where we know that there's going to be so much residual confounding, we wouldn't be confident in the results. So your point is well taken.
Thank you. We all struggle with the same issues.
We do. We have a lot of data, but it's not necessarily better than anyone else's. It's all, you know, we do what we can.
I have another question for you, but I see some people dropping off. And so let me just remind people that you've been kind enough to offer a mentoring session as well. So for those of you that are trainees, that's 1:30, and you can contact Katie Anderson for more information about that. So thank you, Sarah, for your willingness to serve and help us continue to learn together in that capacity. I just wanted to ask briefly about BEST. I believe that's what it's called, the initiative that's sort of the Sentinel of biologics. And I think everybody on here probably knows that biologics are products made from living cells, and that they account for a small amount of overall product volume, but a large and ever-growing fraction of expenditures, and have some very unique toxicities and efficacy profiles. And there's lots we could say about biologics, but can you just briefly opine a little bit about you know, the Sentinel for biologics, and you know, and I guess just a few like any take homes from that. I know you may be only following it at a distance, but I'm just curious if you kind of have thoughts or anything you can share with us about that initiative and how we should be thinking about that based on what you've helped us to learn about Sentinel.
Sure. Can you see the screen, the slide I'm sharing?
Yeah.
This one blank for me.
Yeah.
So the way FDA is set up, I'm in CDER, Center for Drug Evaluation Research. Biologics, or most biologics, are covered by CBER, the Center for Biologic Evaluation and Research. So we are largely separate. So I can't say that I know a lot about BEST. So technically it is part, the slide shows, technically it's part of the Sentinel initiative. I know this is sort of a lot of government speak maybe. So we're over here in the Sentinel system, and ARIA is where I fall. You know, we use the Sentinel infrastructure, which is those tools, the common data model, the data partners. You know, so BEST was set up, it's technically under the Sentinel initiative, but it really does have its own system. And I think that was largely created because of the need for more granular clinical data. You know, a lot of the the information that they need to evaluate, the safety of biologics would be much more granular clinical data found in, electronic health records, both structured and maybe even in unstructured data. And so really claims-based data was not meeting their needs, which led to them developing the system for BEST. So that's sort of at a high level, really all I know. I'm, happy to put you in touch with folks at BEST that I do know that might be able to come speak to that a little bit more. But I know, their focus is right now more into electronic health record and, the much deeper clinical data than the sort of traditional Sentinel can provide. And that was a lot of the impetus for creating BEST.
I mean, I don't know that data is inherently any more, important. I mean, I understand that it may be important insofar as these are often medicines that are administered and dispensed in, institutionalized settings. I don't know. It'll be interesting to see, and I think BEST can learn an enormous amount from all of the hard work that's gone into Sentinel. So we'll see. I think in general, I guess, we're fairly far behind in applying some of the epidemiologic insights and skills and methods that we've developed and have been built largely on the backs of small molecule products to the world of biologics. And so it's a wide open field for anybody that's on the Zoom and wants to figure out where to focus. Lots of opportunities there.
It is. It's fascinating. I know, there are some biologic products that are covered by CDER. So a lot of the biosimilars right now technically are still covered by CDER.
Right.
So we are actually starting to look at those right now, and at least in terms of utilization, although utilization for most of them is still low. But the more complex biologics, obviously all the vaccines is really where BEST comes in, and obviously right now they're quite busy, monitoring vaccines and setting up plans to do that.
Oh yeah.
Yeah.
All right. Well, thank you so much. Any final chance, anybody, if you have a burning question,